So you were talking about slide number 30. Is that correct? So let's look at it together and see if we can figure out what you're. I don't know what you're talking about exactly, but let's scroll down. Work, you piece of. There we go. Okay. 8, 9, 10, 11. Okay, so we're into enzymes. Okay. Okay, so nutritional types of organisms.
That one out because
okay.
You get my text.
Oh yeah. No, I was just making sure that you got it all straightened out, and you didn't have any more questions.
No, So basically, just all living organisms need energy, electrons, and carbon. And so the nutritional types of organisms is just going through the different types of energy, electrons, and carbon, right?
Yeah, there's two types of energy source, right? The sun, which is photo, and then chemical, which is the other one. Okay, just remember that there's two types of energy. There's potential energy, and there's kinetic energy, right? And so we use chemotropy or chemical energy, and we either do one of two things with it. We either store it as potential energy in the case of ATP, or we release it to do mechanical energy, whether that's moving our bodies or we're causing an enzyme to turn, such as ATP synthase. We're taking potential energy and converting it into kinetic energy, which is energy of movement to get something done. And that's really what we're talking about. And then, okay, so based on electron sources, so lithotropies or lithotropes, right, use reduced inorganic substances, and organotropes obtain electrons from reduced organic compounds. So humans, what are we?
We're organic.
Yeah, we're organotropes because we get it from other stuff, and we're organics, right? And lithotropes, so plants would be a good example of lithotropes because they use inorganic stuff like sunlight and carbon dioxide and to create sugar molecules for us. And so they're lithotropes. And that's what that basically means. So inorganic substances are things that we wouldn't consider living, where we as humans and animals, traditionally, we consume living plants, but the plants themselves, they can generate organic molecules from inorganic stuff. And they do that through photosynthesis. Okay. So that answers that question.
Yep.
Okay, so let me hit this really quickly. So really fast. So tomorrow's lab. So what is happening in lab tomorrow?
They're gonna give us already made cultures, and that we have to look at the results of based on that table from the pre-lab.
Yep. So we're gonna do post lab of 5. We're gonna turn in pre-lab and do post lab of 6, and then again, just our own little study group. We do 7. Like kind of read through 7 tonight or tomorrow sometime before lab and be ready to discuss the pre-lab for 7 tomorrow. Does that make sense? And then that way we'll be a little bit ahead of her. But I know that kind of feels like a lot because we kind of got hosed. Normally, you and I we'd just be discussing the pre-lab of 6 tonight. We'd be initially talking about the pre-lab of 7 tonight. And then tomorrow we'd knock out 6's lab. But we kind of got hosed. And so I just wanted to make sure, just know that tomorrow's lab could be pretty intense. Like, that makes sense. It's going to be quick. And like, I recommend that we do, like, for example, one person goes up and gets all the data table stuff for 5, right? Unless she just puts it up. One person get the post lab for 5 done for everybody else, while everybody else starts pre-lab or the lab of 6, if that makes sense. I'm just with you and I just wargaming this because 6, we've got to do bacterial doubling time. And I was looking through this and I'm not really sure, looking at it, how she's going to do this with this, but it looks like we're going to get some cultures, right? So we're going to get the OD of the 600, the cells, and then over the 90 minutes of that lab. So we're going to have one that's at an elevated temperature, and we're going to use the same culture and just leave it at room temperature. And we're going to be doing cell counts over the course of the whole lab over 90 minutes. Well, our lab is like 2 hours long. So if we're not hustling through this, we're going to run out of time.
Yeah.
So that's it. So now I'm done with that. I like to get a gut check on the lab so that we're kind of clear on the procedure, because I feel like it just makes it a little bit faster when you go in there and you haven't done that. it gets you into trouble. Like, you know what I mean? You take time just unscrewing everybody up to get started to do whatever, as opposed to, okay, you two do this, you two do this, set the microscopes up, so let's go, and start to hammer through it. Okay, so let me do this. I actually met with our teacher today, and so I met with the teacher today. Unless you have specific questions about 8, let's just move on.
I do have a few.
Okay.
Sorry.
No, you're okay. Let me go back to it then and bring it back up so we can see it together. Doggone it.
Starting on slide 43.
Okay, hold on, let me get it up again. I'm going to put it up so we can both see the screen together. Just, you know. Okay, there it is.
I don't know if she skipped this or what, but I don't remember like anything about the next 9 slides or so.
Okay. So now I got it up again, and you said it was 43 that you were looking at?
Yeah.
Okay, let me bring it up. And so we're both looking at the same thing, so I don't get confused. Okay. Okay.
Yeah.
So standard reduction potential. I think we're on the same page right now. Okay, so let's look at this guy. And why is this not? Why is it not moving like it's supposed to? Okay, hold on. When in doubt, try it again. Okay. Okay, let me try this one more time. Stupid thing.
Also, I don't think we ever went through the last half of 9.
Okay, no, that's why I'm asking. So, okay, there it's working now. Okay, so that's 43. So standard reduction potential, equilibrium constant for oxidation reduction reaction. So do you know what an oxidation and reduction reaction is?
Yeah.
So what is an oxidation and reduction reaction? That's the first question.
Well, that's the oil rig. So oxidation is a loss of an electron, and reduction is a gain.
Yeah, so that's really all this is showing. So if you look, let's look at the very top one here. So it says selected biological important half reactions. So it says we have 2 hydrogens that are positively charged and 2 negatively charged electrons, and we bring those together. This one's kind of a funny one because normally we associate reduction and oxidation reactions with the movement of hydrogen, right? With that movement of hydrogen. But in the first one, we see it's actually hydrogens that have been oxidized, right? Because they've got electrons added to them, right? And then we have electrons that will then be reduced and given to the hydrogen. And at the end, we're going to have 2 balanced hydrogen dimers together, 2 complete hydrogens that aren't missing anything, that have come together nice and tight, and they're just normal hydrogens that don't want to fly around. What do we call, Laura, just as a little bit of review, what do we call hydrogens like this that are missing an electron? They have a special name. They damage tissues in humans.
I think Addison said this last time.
Yeah, they're called free
radicals.
Free radicals.
Yep, and it's this positive
and negative charge on electrons and hydrogens. When they're not bonded nicely, they go looking for a home, right? They start looking for a place to go to. And what they do is they just go to random tissues and they start to pull, like these hydrogens will start to pull electrons off those tissues and make them unbalanced and kind of poisons them, if you will. And those are called free radicals. Okay, so let's come down here to this second one down. So here we go. We've got NADP plus hydrogen pluses and plus 2 electrons that are minus, right? We do this reaction and we end up with NADPH. We've heard that before, right? We've heard this NADPH. So what's going on here? You tell me what's going on here.
It looks like they're gaining electrons.
So who's gaining the electrons?
The NADP. is added with the H and the electron, so both of them, the NADPH.
Yeah. So again, this is why I don't like it when they show the electrons separate. Normally, this reaction would just be NADPH or NADP and hydrogen, right? And they're separate. And what would happen is NADPH is going to be it's gaining electrons. So what's happening when it's oil rig, right? So this would be reduced, right? So NADPH has been reduced by adding hydrogens and the electrons. And over here, NADPH is oxidized right now. Does that make sense? Or excuse me, the hydrogens are going to be oxidized because they're going to be added to something else. Does that make sense? So that's all this graph is showing. Now look over here, right? So what does it say over here? What's the big word over here at the top of this line?
Volts.
So yeah, this is telling us how much actual electrical energy is created when these reactions happen. So let's look for the one that has the most energy production. Which one has the highest number over here in this column?
The last one.
So yeah, we come down here and look, when we form water, look at how much energy is created by this reaction, right? There's quite a bit of energy going on right there. And so at 0.82 volts, right, is created when we do that. Okay. Now what that also tells us that when we pull oxygen and hydrogen apart in water, how much energy does it take to pull it apart? It takes just as much or more energy to pull it apart when we go to use this stuff for something else. Does that make sense? And then obviously when you see a negative number, that doesn't mean negative energy. You can't have negative volts. Does that make sense?
Mm-hmm.
You can't have negative energy. It just shows an inverse when these are done, right? And so that's what's going on there. So when you look at it, just for the sake of this, without knowing more about electricity and not crossing that bridge, just know that anytime you have a number over here, whether it's negative or positive, it just means that there's energy there. Okay, so 0.42, there's quite a bit of energy here with hydrogens formed in its dimers, right? Let's go down here. Let's find something. Okay, so FAD and H2 forming to FADH2 is 0.2, but look what it says here, raised to the 2. That one's actually squared. Look at this one. Iron, adding electrons to iron, so taking that and adding electrons to iron, it's 0.77 raised to the 3rd. So this one's probably the strongest right here. There's the most energy here with iron. So that's all that's showing. Does that make a little bit more sense now?
Yeah.
It's just showing these reactions, and it's just saying, hey, here's what's going on with these guys.
Okay.
Okay, and then remember the electron tower?
Yeah, sort of.
So what's going on is it's saying, hey, man, these guys up here are better electron donors. They want to give stuff up. They want to give electrons up, and these guys down here want to take electrons. And that makes some sense, right? Because here's water and oxygen down here at the bottom, right? And we know that oxygen at the end of oxidative phosphorylation, what's the final electron acceptor?
Oxygen.
It's oxygen, right? So if we come up here and we find these two hydrogens up here, they're like 0.42, negative 0.42. Those guys drop, and they're creating all this energy when they bond with oxygen. So there's a huge jump there, almost a jump of 1 from negative 0.4 all the way down to 0.8 positive. So it's almost 1 whole volt complete jump. And that's all it's showing. It's just saying, hey, man, the further apart these things are, the more energy they create. Okay, let's see. Yeah, so there's like, nitrate, nitrites right here. You got like fermate, pyruvate and lactates up here, FADH2, acetate. I mean, you kind of look through here and you can see what's going on with these guys. So anything else you want to add on that?
No.
Okay.
Okay, so
carbohydrate catabolism.
So this is just talking about the ways to produce ATP, and it's cellular respiration, and that's aerobic or anaerobic.
Yep.
And then fermentation, which is also technically anaerobic. That's all that one's saying, right?
Yeah, and then just remember, so when humans, when we don't have oxygen present, what do we go to? Do we go to anaerobic respiration or do we go to fermentation?
Anaerobic respiration.
And we go to fermentation. lactic acid and the production of lactic acid in humans is considered a fermentation. It's anaerobic, right? Because there's no oxygen present. And that's the tricky part of this, okay? So remember, cellular respiration, we do anaerobic respiration. And then when we don't have oxygen present, it is an anaerobic reaction. See right here? It is anaerobic, but humans and plants will go to fermentation. When plants don't have oxygen, what do they create? What's the end state for plants when they ferment or they don't have oxygen present?
We just talked about this. Acid? No.
Well, they create grandpa's cough syrup, right? They create the bean of the Mormon church. It's one of the Mormon word of wisdom things. Ethyl alcohol. When plants are denied oxygen, they create ethyl alcohol, and we call it fermentation. And so in plants, anaerobic fermentation creates ethyl alcohol. In humans, when we deny ourselves oxygen, we do anaerobic fermentation, and we create lactic acid as the byproduct.
Okay.
Okay. And I believe she talked about in class that anaerobic respiration is reserved for like bacterias and stuff. Okay. Let's move through the slides and find it. I think it talks about it right here.
Oh, yeah.
So all cells undergo glycolysis.
So I get the next three.
Okay.
I think I'd have to, yeah. So I'm good till there.
Okay, just anaerobic respiration. The final molecule is inorganic stuff. Fermentation, it's organic molecules for fermentation. Okay, and it happens inside the cells. Comes from inside the cells. Okay, this one here. And if you can't explain this, I mean, I don't know what to tell you.
Yeah.
This slide here, you should be able to do this one in your sleep.
Yeah.
So let me ask you this question. C6H12O6, what is this thing? What do we also call this? What?
Glucose.
Glucose, yep. So 6 glucose and 6 oxygen. And all the steps of oxidative respiration or oxidative phosphorylation, these are the final components through all these steps, okay? Now, what is the energy that's created at the end of aerobic respiration after all the steps of glycolysis? Form acetylcholine,
ATP.
Krebs, yeah, ATP, water, and CO2 are all the products of this whole process.
Okay.
Okay, awesome.
Okay, we can skip now. Oh,
okay,
wait.
do you remember the Ebden-Meyerhoff-Parnas pathway? And then
let's go over that actually.
Yeah, let's go over those because I think we do Ender-Meyerhoff's ourselves, like in humans.
Yeah,
let's just double check. It should talk about it.
That's what she said.
Yep, Ender-Meyerhoff is us. So let's skip through here until we find the other ones.
It's slides 53.
Okay, 53.
53 is where the Ender-Dudoroff pathway is.
Okay, so we got Ender. So let's look at this one. Used by some prokaryotes, replaces the first phase of Ender-Meyerhoff pathway. So let's see how it replaces. Oh, here it is. So in ours, in the first step of ours, Ender-Meyerhoff, what happened to glucose?
It got broken down into 2-pyruvate.
The 2-pyruvate's correct. And this one, an Ender-Dudoroff or whatever, it's created. What happens to it? It becomes glucose 6-phosphate. So it just physically adds a phosphate to a glucose. And then through that process, ADP plus pulls a hydrogen off and becomes, well, it becomes NADPH and then an H plus with it. And then it becomes 6-phosphogluconate, right? And it doesn't get broke. It's that thing, it stays a 6-carbon molecule all the way down to pyruvate. So that thing doesn't lose any carbons at all, and no CO2 is produced in that one at all. And it keeps going down to, okay, so here it becomes pyruvate and glyceraldehyde 3-phosphate, and then glyceraldehyde 3-phosphate drops down through this. And look at how much stuff it creates. When from one glucose molecule, how much ADP and NADPH is it produced?
Just 3 total.
Yeah, it's really not very productive.
When we're doing our normal 4 cycles, like the glycolysis and the formation of acetyl-CoA, the Krebs cycle, oxidative phosphorylation, the glycolysis cycle that we use is the first one, right?
Yeah, enter Meyerhoff.
So then these other two are just alternate pathways, and this one we're talking about right now is for some prokaryotes, and the other one is, what is the other one for?
So let's see if we can find it, because it should talk about it in here somewhere. Here it is. So pentose phosphate pathway produces sugar in NADPH. And then let's see if it tells us who does this.
Because I'm just confused when we use which one.
Well, and I think she told us that she showed us these, but we don't have to know all the steps in these. We have to know ours. Embedden Meyerhoff or whatever. Okay. So she wants us to know what these are, but she didn't expect us to know all the details of these, because these are quite complex. Okay. They're pretty doggone complex.
When we use it, but
yeah. So what I think what she was saying is she's introducing us to us under the guise of just continuity. Like, she wants us to know this because it's fundamental to microbiology.
Yeah. I wrote down that every cell does this pathway. I just wasn't sure when they do it, but she probably didn't say, so that's okay.
Yeah, I'm just saying, but what she's saying is, like, so you and I want to be nurses, right? In the sense that we wouldn't have to really know this stuff, because as nurses, we're not going to be really dealing with it. We just didn't know that it's there, right? Mm-hmm. But say you and I decided that one day to go on to get a master's or a PhD, and we did chose to go a microbiology route. Say we wanted to do research into into bacteria. We then may be getting into this more, because those organisms use these pathways more often, and then we'd be digging down these and really learning them.
Okay.
But yeah, they're just different pathways of how different organisms break down glucose, right?
Yeah.
And it just depends on how they do it. We already know the most productive or the most efficient that produces the most energy is the Embedden Meyerhoff. That's the one that creates the most energy.
Okay. All right.
And now we're back to our own stuff again.
Yeah, I'm good for the rest of 8, so we can go wherever you were wanting to go.
Well, no, I'm just asking. So let me turn this thing down. And let's move on. Oh, did I just kill something? Oh, sorry. Okay. So let's look through 9 really quick. Let's just look through 9.
I think we made it most of the way through.
Okay.
But we stopped. I think we stopped right when we got to the culture stuff.
So did she cover all this in class? Okay. So I want to make sure, Laura, that you understand this concept. You seem to have some issues with this last time. I just want to make sure that you understand this, okay? So hypertonic and hypotonic solutions.
Yeah, okay. Hypotonic is when it gets water or fluid in the cell and it makes it like blow up like a balloon. And if it pops, then it's osmotic lysis. And then hypertonic, the water leaves the cell and plasmolysis can occur.
So the way that I explained this in the pathophysiology class, and let me see if this helps you a little bit, okay? So you have a cell, right? The cell, whatever cell it is, eukaryotic, prokaryotic, or whatever, right? And say that your cell has 5 red Skittles in it, okay? Just say, using Skittles, like some candy that you understand, has 5 red Skittles in it, inside of it. And so all the time we're talking about the one cell always has 5 Skittles in it, okay? So what happens is the Skittles themselves can't physically move in and out of the cell, but you do have water with your Skittles that can move. So what happens if I put the Skittle cell with 5 Skittles into water that has 10 Skittles in it? What way does the water want to move?
I have no idea.
Okay, so let me take a step back.
Your Skittle example.
Okay, let me take a step back. Just use any. So, say I have a cell that has 5 molecules of water and has 5 red Skittles in it. What is the proportion of water to Skittles inside the cell?
It's equal.
It's 1 to 1, right? When I put 5 over 5 and divide them, I get 1 over 1.
You want the water to go out.
Yeah, I'm trying to get the same ratio of water to the number of molecules. So now, what if I take and I put that same Skittle into solution that has 10 red Skittles and 10 molecules of water with it? Well, right now, right now I have it's equal. What we're always trying to do is we're trying to get proportion, right? We're trying to get this to proportion. So we have 10 to 10, but we have 5 to 1, and they want to be in proportion. So what we're trying to do is always get the number of whatever molecule or structure we're looking at to the same number of water that it's in. And that's why this stuff moves the way that it does. That makes sense? So when you look at this, so the number of water molecules is unlimited. That's the problem, is that we can't give us a certain number of water molecules. So the water molecules are unlimited, but they're isotonic. They'll move equally to level out and get the same proportion of water molecules to whatever we're looking at. So say salt in the cell. Well, okay, so now I have a molecule with 5 molecules. I have a cell with 5 molecules of water and whatever proportion of water it needs to be balanced, right? Well, now I put that same cell in a hypertonic solution that has 10 molecules of salt. Well, the salt molecules can't move across the plasma membrane freely, but the water can. And so there's more concentration of salt outside the cell. And so the water inside the cell moves outside to try to dilute the salt that's outside the cell to the same concentration as inside the cell. And that causes it to get small, right? That's why it gets smaller. Now, what if I reverse that and I have a cell with 5 molecules of salt in it and I put it into a hypotonic solution, meaning there's only 2 salt molecules outside the cell? Well, in that case, there's more salt inside the cell and water molecules rush inside the cell because it wants to make the concentration of salt inside the cell the same as the concentration outside the cell. And because the salt molecules can't move, the water molecules are what's moving to try to create that balance. That's it. And I'm sorry, that was probably a horrible example. I apologize.
No, that was fine.
That's the thing that I think when we talk about hypotonic and hypertonic and isotonic solutions, we don't talk about the problem is that, you know, normally that salt would move, right? Normally it would be the salt that would move across that barrier to get that balance back, right? But because the salt can't move, that's why the water is moving. So just remember, hypertonic, I have more molecules of something outside the cell than inside the cell, and that's why the water inside the cell is moving out. It's trying to bring the concentration of that particle outside the cell to the same as inside the cell, and that's why the water is moving. And then again, on the opposite, when I do hypotonic solutions, that means the concentration of any particular molecule, say this kind of time it's calcium. Say I have calcium inside the cell, and I have 10 inside the cell and 2 outside the cell. Well, then water from outside the cell rushes in to try to reduce the concentration of calcium inside the cell, and it's using water to try to create that balance, and that's why it does that. Okay, so on hypotonic solution, if I have 10 calciums inside the cell and 2 outside the cell, water is going to aggressively rush into the cell, and if there's too much water coming in, what does it do to the plasma membrane eventually?
Worse.
Yeah, it ruptures by getting so large, it's like blowing a balloon up too much, and eventually the balloon just explodes, right? Because it can't handle it.
But the one that's
a little bit creepy is this hypertonic plasmolysis. Can you tell me what's going on with this one and what's going on? This crazy one.
Which one?
When plasmolysis. What's going on in plasmolysis? Why is this so bad? Well, let me so I can use an example. Do you have curtains in your house anywhere that are hooked to a curtain rod?
Yeah.
Okay.
It pulls the wall in with it because it's connected.
Yeah, the plasma membrane is like your curtain, right? And it's hooked to the wall by a curtain rod, correct? And the curtain rod is hooked to the wall of your house by a bracket, a metal bracket of some kind. Well, when you start putting pressure on that curtain, the bracket and the wall of your house are not going to move, right? They're going to stay in place. So the curtain starts to tear because the wall's not moving, but your curtain, you're putting pressure on it. So what's really happening here in plasmolysis, you're just putting pressure on that plasma membrane, which is pulling on the cell wall. Well, the cell wall on a bacteria is stronger than the plasma membrane. And so as more and more water rushes out, it puts more and more and more pressure on those connection points where the plasma membrane is connected to the cell wall. And eventually, because of plasmolysis, it's shrunk so much, it's put so much pressure on those plasma membrane connection points that eventually it tears it open.
Okay.
Is that clear as mud?
Yeah.
Okay, so you tell me what's going on then. I want to hear you explain it. And I wish this cell wall thing here, this graph, I wish it showed these, because see, it showed that plasma membrane completely disconnected from that cell wall. It'd be nice to have a couple of points where it stayed connected, but it doesn't do that.
So the plasma membrane has so much water that leaves that it starts to get small, but because it's connected to the cell wall, it rips away from the cell wall, and that's bad.
Yeah, perfect. That's perfectly what it is. And that was so boring. It was awesome. You look so excited right now. The look on your face is like, why the hell are we still talking about this? Okay, hemophiles. Okay, water activity level. What's going on with water activity level? Can you explain this to me?
I'm trying to find where you are.
I'm on slide number 14 and number 9.
So that's just talking about the water that's available. So if you freeze dry, yeah, good job. If you freeze dry something, or you give it a lot of salt or sugar, then the bacteria can't grow because they don't have enough water.
Perfect. No, that's exactly right. Yep. So like she gives the example of honey and beef jerky, right? So in the case of honey, we've displaced all the oxygen, or excuse me, we've displaced all the water with what? What's displaced all the water in honey?
Sugar.
Yeah, sugar's displaced it. So it's not leaving any water left for anything growing there. In the case of beef jerky, we're using 2 processes. We're actually physically sucking the water out of the beef jerky packet by actually vacuuming it shut. And then we're also doing what to it?
Giving it salt.
Yeah, so salt and water in that case. Have you ever opened a beef jerky packet and found that little white packet, that little white bag inside there? And what does it say on the front of it?
Don't eat.
Don't eat. Why does it tell you not to eat that?
Because it's bad for you.
Well, it's not that it's bad. It's because that's a highly water absorbent chemical in there. And if you eat that, what is it going to do to your system when it goes through there? It's going to absorb water in your system, and it's probably going to get enlarged as well. And I would imagine that the chemicals in that probably aren't, they're highly concentrated, and they're probably not very healthy for us. I'm sure you could eat that and be okay. You might get a little sick. I mean, obviously don't eat a lot of it, but yeah, it's going to dehydrate you as it moves through you. And it's going to, you know what I mean? It's going to be sucking up your water. And so it could cause you to maybe be constipated or have like funny urine or something. You know what I mean? And so that's, I don't think it would probably kill you. But maybe for a child, though, if a young child ate one of those little packets, it might have a more significant impact on a kid because their body and volume of water is quite a bit smaller. So that's it. That's what's going on with water activity. I think we beat that one up too. Okay, carbon. I'm not even talking about carbon. Nitrogen, trace elements. So we have cofactors and coenzymes. What's a cofactor?
I know this.
So the word coenzyme, both a coenzyme and a cofactor do the same thing.
It's the non-protein part of an enzyme.
Yeah, well, it's a inorganic substance that binds to a different part of an enzyme that changes its shape and allows a different chemical reaction to happen. That's what a cofactor does. A coenzyme does the same thing as a cofactor. It binds to another enzyme and it changes its shape, but an enzyme is just another protein that does it. Do the exact same job. They just do it, and they do it the same way. One just happens to be another enzyme or a protein, and the other one is just an inorganic molecule like iron. Like, remember we talked about our blood? So iron in hemoglobin is a cofactor to hemoglobin, and it facilitates oxygen being bound up to it so it can move it throughout the body. That's a cofactor, and a coenzyme is something that does different jobs, but it uses proteins to do all that stuff and make it so it's available to do its job. Okay, we beat that one up.
Yep.
Okay, let's talk about oxygen and bacterial growth in a totally different way than she did. let's look at the base words, okay? I think we both know what aerobic and anaerobic is. That makes sense. Like we know that aerobic is oxygen present and anaerobic is oxygen not present. That part we know, but let's break these down and let's look at these, okay? Okay, so obligate. So if I'm obligated to do something for you, what does that mean?
You have to.
Yeah, I have to do it, right? I'm obligated. So, like for example, I have a home and I have a mortgage on that home, and by contract, I'm obligated to the mortgage company to pay the mortgage every month. That's an obligate responsibility that I have. I have to do it. And so if I have an obligate anaerobe, what does that mean?
That it has to have oxygen.
Yeah, it requires oxygen to have it. If I have an obligate anaerobe, what does that mean?
Has to not have oxygen.
Yeah, oxygen cannot be present for that bacteria to grow. Now let's look at the other ones that are over here. So what's facilitative mean?
Run, to organize, sort of.
So let's use an example from school, right? We have a teacher. Do you have to learn if you have a teacher? If you go to college and you sit in class and your teacher shows the slide decks and dispenses knowledge, do you have to learn? Do you have to learn what they're telling you?
No.
No, they facilitate. They're mentors, right? And vice versa. You can go to a teacher and have a teacher who's a total crappy teacher, and you can still learn the knowledge for that class, even if the teacher isn't that good. But the teacher is facilitative. Okay. You don't require, so a facilitative anaerobe does not require oxygen, but it does grow better in its presence, right? So you learn better in a classroom with a teacher teaching you. But could you still learn microbiology from YouTube clips and from a textbook that you bought on your own?
Yeah.
But is it easier with a teacher present and someone to mentor you and help you?
Yes.
Okay, so that's all I'm saying is that sometimes it's just easier to look at the words we don't know. So facilitative anaerobe just means, yeah, it doesn't require oxygen, but man, it sure does grow better when it's there. Okay. What about aerotolerant anaerobes?
They can do either.
Yeah, so aerotolerant means yeah, they tolerate oxygen, but they don't need it. So they grow with or without O2 present. So aerotolerant just means that they can tolerate oxygen. It doesn't need it, doesn't harm it, doesn't help it. And then what about microaerophiles?
Micro needs a little bit, so it needs a little bit of oxygen.
So what's really, really special about microaerophiles? How much oxygen are we talking about?
2 to 10%.
Okay, so let me ask you a question. In ambient air right now that you and I are both breathing just in our houses, how much oxygen concentration is in the ambient air that we breathe?
Well, a lot.
It's like, I think it's like 18 to 20%. So do micro aerophiles grow well in the house?
Yeah.
No, they don't, because the concentration of oxygen is too high for them. They don't like your house. They got to go somewhere where they can get a little bit of oxygen, but they can control how much they're getting. And the ambient amount in normal atmospheric air is too high for them. That's why they're called micro aerophiles. Okay, so just remember that's what it's talking about. That those guys have very, very special environments that they live in because ambient air in the earth is too high oxygen concentration for them. Okay, and then we look at these guys right here. So there you go. So for a good example would be like microbes that live in seawater, but live in say 2 to 3 inches of seawater. They stay at 2 to 3 inches in seawater. They have access to some oxygen, but because they're all completely submerged, they isolate themselves from ambient air. They're not completely exposed to it. Okay, I'm done beating that up. Do you want to explain these to me? These test tubes?
What?
Just the next slide. Slide number.
Yeah, no, I got it.
Yeah, just slide number, whatever. Just
The
walk
first
through them and
one,
just so
they
you.
need oxygen, so they're living close to the top. The second one, they don't need oxygen, so they're living at the very bottom away from it. The third one, they can have some. They do better with oxygen. than without, but it can do either. And the fourth one can do either. And
they're ish. They're like ish. I can have it. I don't have to have it. Whatever. I'm fine either way. They're ish. They're ish microbes.
And then the last one just needs a little bit, so it's not all the way at the top, but it's close.
Okay. Oxygen, easy. Let's see. Look at bases of different oxygen sensitivities. Oxygen easily reduced to toxic reactivity. Oxygen species. So what's going on with this one? Slide number 23.
It's just talking about how much oxygen it has.
Yeah, it's just saying. So again, if you look at the oxygen hydrogen concentrations of what they're talking about, these different makeups, and then also look at the electrons, right? So again, these are oxygen concentrations and electrons, and so we got like 2 oxygens here, but it's raised to 1 thing. It makes sense. So that's what they're talking about. They're just talking about the reactivity of these guys, and it's really a relationship between oxygen, hydrogens, and these electrons, and that's all that's going on. And then the next one just shows what's going on. So superoxide free radicals. So O2 minus plus O2 minus plus 2 H's. Superoxide dismutase creates hydrogen peroxide and 2 oxygens, 2 dimers of oxygen. And it's just showing these reactions. Catalase is cutting them apart. Perioxidase is cutting them apart. And then, okay, here's the fun one, biofilms, dude. Biofilms are awesome. Okay, so what's going on with biofilms? So let's ask ourselves, what makes up biofilms? What are they made of?
Microbes.
So biofilms, well, the center of them is microbes, but what biofilms are really doing is they're protecting microbes. They're protecting something inside. They create a really, really nasty film to protect what's in there. So in this case, polysaccharides, proteins, and waters.
Yeah.
That's what's going on. So they protect the microbes. And what's the medical importance of these guys?
I know we talked about this, but I didn't write it down.
It's okay. We're going to do it now.
So
if I put a sugar, protein, and water layer around something, is it going to be protected or is it going to be really weak?
What was the question?
If I go and I throw a polysaccharide, a sugar layer, a protein layer, and a water layer around something, especially in organic stuff, am I making that thing really, really hard to get through, or am I making it easy to get through?
Hard.
Yeah, it makes it really, really difficult. So what happens is bacteria and different things get embedded in this biofilm, and it makes it extremely difficult to kill them, because we got to first get through that biofilm to get down to down to the bacteria that we're trying to kill. So again, in humans, and you're going to learn this when you get into pathology, the way that we treat a lot of these things is we'll go in, we'll irrigate the wound, like completely drain it with water and stuff. And then what we'll do next is we'll actually go in there with dry gauze or dry cotton cloth, and we'll stick it inside that wound. We'll stick it inside of the wound itself, and then we'll draw it out, because what it's done is it's drawing that biofilm with it. We irrigate it to get any loose microbes and biofilms off of it and try to drain those out. And then we pack that wound with that gauze and that cotton and that cotton bandage, and then we draw it out after a while, and it pulls all that biofilm and again more and more of the bacteria out, and now we just have less to fight because we've mechanically drawn it out. That's the start of fighting this stuff. And then we're trying to use we're trying to use antibiotics to kill this stuff, but we're doing the best that we can to get that dang biofilm off of it before we start. So it's just very, very energy intensive. It makes it very difficult to fight these guys. Okay, we cool?
Mm-hmm.
And this guy, are you looking at the slides, man, right here?
Yeah.
It's showing an abscess, man. This is an abscess tooth. That's what that actually is. So that yellow and pussy green stuff, that's abscess in a tooth. So if you ever get an abscess on a tooth and you're getting a yellow pus from it, you'll know what it is. It's the biofilm coming off the bacteria on an abscess in a tooth. Yay! And this here, look at this right here. This is actually a stage 4, like bed sore in here. You got skin layers and muscle layers here, and they've cut down and dug into it. And you got all this necrotic tissue underneath it with all that nasty gummy crap. That's all biofilms. Ooh, that looks so awesome. Dude, aren't you excited to be in healthcare? Okay, so the three stages of biofilm are what?
Attachment, growth, and detachment.
Yep, attachment, growth, and detachment. They go off and find somewhere else to go. Okay, and then this one here just shows, again, just how the biofilms are formed and where all the stuff, the different microbes are going to be embedded in here, depending on what environments they like. That's where the microbes are going to be, and that's what we learned. That's what we learned. Those, facilitated anaerobes, facilitated, aerobic, that stuff, and that's why they're going to be embedded where they are. That's it. So guys that hate oxygen will be right in the middle of the biofilm. Those that need more oxygen are going to be on the outside. Any questions on that, guy?
No.
Okay, awesome. Okay, what is quorum sensing?
Well, cancer doesn't have quorum sensing.
Nope, cancer definitely does not have quorum sensing. So what does it do?
It's like knowing when there's too many cells.
Yeah, in normal society, if you have a room with 10 chairs in it and all 10 chairs get someone's butt fill in it, what does that mean? It means the room is full, right? It means no one else can come in there anymore. Quorum sensing is the process that normal cells use to tell them to stop growing and stop dividing, right? Because we're full. The space that we have is full. But cells like bacterias and viruses and cancer and stuff, they lose their quorum sensing ability, and so they continue to divide and continue to grow, forcing good cells out of their way. They continue to just do that, okay? And so the one thing that we didn't talk about as much is, so what'll happen is, right? So you have an R protein, and you have the R protein, and this basically an inhibitor, right? Right now, you have these protein molecules out here. There's only 3 of them in low cell density, or AAL, low cell density. And so there's not enough of them. None of them are hitting this protein, this R protein, to tell them to stop growing. But as those start to divide, more and more and more of them fill that same space. And what happens is those molecules of bacteria start to bounce into that R protein. It attaches to this inhibitor, and then that inhibitor says, "Hey, man, stop growing, stop doing this." And that's all that's going on. It just tells that's actually the nucleus. I'm sorry, that little thing with the stick on it is the nucleus. So it's basically this R protein attached to the nucleus. It says, "Hey, man, stop growing," because we're getting too many of these targets, these auto-inducers on us, that they keep hitting me and telling me there's a lot of them. It means it's time to stop growing. And that's all that quorum sensing is. And I think it's not really difficult to understand that. Okay, awesome. Okay, culture medias. Did we talk about these?
No,
Okay, well, let's talk about before we look at all this, let's talk about the culture medias that we've seen so far in the lab. What have we seen in the lab so far?
E. coli.
Well, not specific bacteria that we've dealt with, but what have we dealt with? To draw those specimens out and look at those on the microscope. Where did we get those specimens? Where did we get those cultures from?
Somebody did them.
Well, no. When you go to the lab, they're sitting in a test tube, right?
Yeah.
They're in a test tube, right? And there's a material that they grow in or on. In the case of the stuff that we're working with, it's a physical, solid culture media we call agar, right? It's that clear liquidy green crap. That's a physical culture media. It's a solid physical culture media that those things like to grow in. Well, what would be a physical liquid culture media that we could use? You ever heard the
I don't know.
well, agar, we can heat agar up and make it liquid and whatnot, but agar at room temperature likes to be solid. I'm talking about broth. In the lab, and I'm sure before this is over, we'll work with some broths, that the bacteria and the medium that it's in is in a liquid format all the time. and that they call that a broth. When it's liquid all the time, it's a broth. When it's solid, it sits solid at room temperature all the time, and that traditionally is the agar. Okay. So chemical composition, what is its chemical composition? So some agar is agar. It's like animal matter augers. Some of them are blood augers. Some of them have chemicals that we add to them to do different things to get certain cultures to grow. So like their function is reducing, selectivity, differential, enrichment. We're going to learn about these 4 things, but the kinds of media that we use, the kinds of culture media that we use is going to facilitate. So if it's a selective culture media, what would selective mean?
That it can't hold everything in it.
Well, yeah. So what it's saying is that we're going to put our culture media together and we're going to put some selective components in it. So when we put a bacteria in there or a culture sample in that agar, if we're selective, it means we put things in the culture media to selectively force certain things to grow and to inhibit other things from growing. Does that make sense?
Yeah.
So we're trying to selectively grow certain things, and we can design media cultures, our culture media, to help to select what we want, right? Differential, enrichment. Does that make sense? So enrichment is, hey man, we add more food for bacteria to grow. And so like in the case of, we could enrich an agar or a solid media by putting glucose in it. We could put an extensive amount of glucose inside of our agar media. And so we're enriching that agar to make it more conducive for bacteria growth, because a lot of bacteria do like sugars to eat. They love them. And so if we add more sugar by enriching it, it makes it easier for them to grow. Okay, let's look through these really quick. Okay, so agar we talked about is a solid, right? Okay, agar is used, it's red agar, it's used to solidify liquid media. It usually sits solid at room temperature, and it's good to isolate and establish pure cultures. That's what we like to use it for. So there's plate agar, deep tube, broth, right? We talked a little bit about broth, agar slant. Okay, and remember, I don't know why they're calling broth. It's showing broth in here, but broth is usually a liquid at room temperature, and so it's not technically considered a solid agar.
No, that is a liquid.
But is it? Yeah, so it's not a solid media agar is what I'm saying. In this slide, it shows it right there in the middle of those, a deep tissue or deep tube and slant agar. It shows it next to those, and in this slide, it's talking about solid agars. So whatever, let's just keep going. You know what I'm talking about. Any questions on that?
No.
Okay, chemically defined agars, all components and their concentrations are known, right? So like we add glucose to it, ammonium sulfate, sodium chloride. So basically we're adding components to it, and we know the actual amounts of the stuff in there. And by knowing that, we also know how it's going to help affect, biological growth. It's going to help control and show us how it's growing. That's chemically defined or known stuff.
Okay.
Is that good? Okay. Complex media contains some ingredients.
Complex media.
What was that?
I said I'll tell you about complex media.
Okay, you tell me about it then.
I will. So you don't know all the components. Like beef extract could have a bunch of different things in it, but you don't know what's in it. And that's basically
it's beef extract. It's animal stuff, right?
Yeah.
So yeah, no, you got it. It's just unknown. It's an unknown how much other crap is in it. Yay. Yay, beef extract. Okay, so here we go. Reducing selectivity, differential, and enrichment. So anaerobic cultures, reducing media used to contain and to get rid of O2. So in this case here, the reducing agent is these methylene blue, or not methylene blue. It's this envelope containing sodium bicarbonate and sodium bromohydride, and that stuff. But when that chemically reacts, it forces the oxygen out and replaces it with carbon dioxide and sodium and different things, and that displaces it, and that reducing media gets rid of that oxygen. So remember oil rig, right?
Yeah.
Oil rig, reducing. So it's getting rid of the oxygen by reducing and getting rid of it. Okay, cool. Okay, here we go. Anaerobic chamber. Yay, airlock. Awesome. Selective media. Favor or select for growth of some microorganisms and hit the other ones, just like I said. So this one, it's called McConkey agar. It's selecting for gram-negative bacteria. So the media itself is designed to inhibit gram-positive, but it accentuates and enhances gram-negative growth in this agar.
Hang on 1 second, okay?
Hey, it's 5 minutes to the hour. Is it cool if we just end right now? Are you good with that?
Oh, if you want.
Yeah, I need to use the restroom at all.
Okay, great. We can stop.
I need to pop smoke and use the restroom. And I was kind of holding on to get to the hour, but we've been going for 55 minutes. I'm gonna be okay. I just need to go.
All right, let's call it then.
Thanks, my dear.
Okay, bye.
Later. Bye.